Hello, my name is Pierre Forze. I'll be covering the material for this section on the course, uh, which will involve studying neuropathophysiology. There's a PowerPoint that's provided as resources, and the PowerPoint has slides that include an image, and below the image are notes. The notes are designed to provide the necessary information to guide you to what you are responsible for in this course. So, for example, many of the slides have images that have a lot of details, many more details than you need to know for the exam. So the notes that are under the image give you an idea of what parts of the image are important for you to understand. So the objective of this course will be covered by first an introduction. It will be a brief introduction, which will give you an idea of the organization of the nervous system and some key points that you should have already covered in previous courses. So simply as a review, this figure is a flow chart of the nervous system, just so that you remember that the central nervous system is the brain and spinal cord, and this is where all of perception and voluntary actions are initiated. So for perception, it requires sensory receptors, which are outside the central nervous system. So we need a way to bring that information to the central nervous system for perception. And so you have the sensory division, which has all of the receptors, and they transmit information through the peripheral nervous system, which are the nerves, to the central nervous system, either at the level of the spinal cord or at the level of the brainstem. Now, the central nervous system will use the sensory information to control our behavior. It could be movements. So to produce movements, the central nervous system sends signals to the peripheral nervous system, that is through the motor neurons, which reach the motor division. And in particular, the destination is the somatic nervous system containing the skeletal muscles that will contract to produce movement. The central nervous system not only controls limb movements, but it also controls the internal organs. So that is done here again through efferent projections in the peripheral nervous system that go through the motor division, but form part of the autonomic nervous system, which includes a sympathetic division and a parasympathetic division. We'll cover these in greater detail later. Now, just to review the anatomy with you, this is the spinal cord. So the spinal cord has at the bottom the sacral area, the lumbar, thoracic, and cervical. And as soon as the tissue, the nervous tissue, enters the cranial vault, then it becomes the brainstem. So inside the cranial vault, we find the brainstem, which includes the medulla, the pons, and the midbrain. We also have behind the brainstem, we have the cerebellum. Up above the brainstem, we have the diencephalon, or the thalamus. In this case here, it's deep colored, somewhat purple. And then overlying all of this, you have the cerebral hemisphere. And between the cerebral hemisphere and the diencephalon, we have the basal ganglia. So we'll cover the functions of all of these regions in this course. The next part is to talk about connections. These are the principal functions of neurons. So neurons are the basic units of the nervous system. They allow for function of neural networks. And a simple example is shown here as a reflex arc. So you would have a stimulus that is transduced in electrical activity by a receptor, and then this, through synaptic chemical transmission, is propagated through a network. In this case, it's a small reflex network that leads to an association neuron that projects to a motor neuron that communicates with an effector. For example, it could be skeletal muscle or cardiac muscle or the intestine to produce a response. So this is a very simple network, but it shows the principles used by the nervous system. That is, it's the connection of neurons, how they're connected, that leads to the function of the nervous system. We do not know all of the connections of the neurons. It's extraordinarily difficult to identify them. There's a lot of connections. The nervous system contains 10 to 50 billion neurons. But we know for a fact that it's the way that the neurons are connected that determines the function of the network. The next figure is the start of our study, and it's to describe the sources of disease. Now, we're going to describe at this moment the source of all diseases, and we'll come back later to describe them in detail. So I'm going to follow with your notes. These are the notes that you find under the image in the PowerPoint. So for disease, there are many ways to disrupt the activity of neurons. So one example would be trauma. So you can have a physical trauma. For example, you can have a section of the spinal cord, and that will produce paraplegia if it's in the thoracic region, or quadriplegia if it's in the cervical region. Another is that there are congenital birth defects. So these are multiple environmental and genetic factors that can lead to developmental deficits. So one example would be spina bifida. In spina bifida, what happens is that the vertebrae, which normally protect the spinal cord, do not develop properly, and they're not protecting the fragile spinal cord. So the spinal cord will get damaged. It's through normal movements during development of the child, there will be some movements that cause damage of the cord, and there will be consequences to that. Another example would be hydrocephalus. In this case, what happens is that the cerebral spinal fluid, instead of the volume that is produced being evacuated so that there's always a fixed volume, what happens here is there's a blockage. So the fluid accumulates and compresses neurons inside the cranium, and that's going to cause damage. Another example would be cerebral palsy. And in this case, there's abnormal development of the motor control areas. in the cerebrum. So the person would have abnormal control of movement. Another way to damage neurons is through genetic disease. So, for example, Huntington's disease is a genetic disorder. And in this case here, the genes have code that indicates that at a certain age, when the person is in their 20s or 30s, that the basal ganglia should start degenerating. So cells in the basal ganglia are programmed to die around 25 to 30 years old. And since the basal ganglia is involved in the control of movements, the person will have abnormal involuntary movements. Another example is amyotrophic lateral sclerosis, sometimes called Lou Gehrig's disease. So this is another genetic disorder where the genes tell the upper and lower motor neurons to degenerate. So the upper and lower motor neurons, which are responsible for the control of movement, degenerate. So the person becomes paralyzed and will die largely because of difficulty in breathing and coughing when there's an irritant in the lungs. Another method is through the immune system. And here, an example would be myasthenia gravis. Myasthenia gravis is a disease where the antibodies attack acetylcholine receptors on skeletal muscle. So our own immune system attacks them and gets rid of them. So now the acetylcholine that's released to stimulate the muscle has no effect. So the person is paralyzed. Another one is degeneration for unknown reasons. So for example, in Alzheimer's disease, we don't know why, but half of the cortical neurons are lost. They degenerate. Another one can be an infection. So you can have, for example, poliomyelitis. This is a viral infection. The virus enters the body and is attracted to the motor neurons. So when it sees motor neurons, it will enter the motor neurons and reproduce, which will damage and kill the motor neurons. So since the motor neurons are essential for voluntary control of movement and for reflexes, the person becomes paralyzed. You can have meningitis, which can be viral. Viral meningitis is not so dangerous, but bacterial meningitis is very dangerous because the body has a lot of difficulty protecting against bacteria in the nervous system. So this typically, this leads to loss of function. The meningitis is an attack of the meninges, but the meninges are directly on top of brain tissue. So when the bacteria attacks the meninges and the immune system tries to attack the bacteria or the virus, that can spill over on the neighboring neural tissue, and you will see damage, the effects of damage to the neural tissue. The person might have loss of arousal, or they'll sense a loss of sensation or of muscle control, headache, nausea, things like this. Another one can be neurosyphilis, which is a bacterial infection, and this bacteria kills neurons forming the dorsal roots. So what happens is that you will lose sensory function. So tabes dorsalis will be an example. You can have a tumor. Now, tumors are derived only from glia. They're not derived from neurons because neurons cannot divide. Neurons are protected from division. That's maybe an advantage for cancer, but it's a disadvantage for repair. Neurons do not repair. If you irreparably damage a neuron, that's the end of it. But there are supporting cells in the nervous system called glia, and those can divide to repair, but they could also cause tumors. Other sources of disease are related to the vascular system. So you can have an ischemic stroke. That is due to blockage of a vessel. Or you can have hemorrhagic stroke. That is due to a ruptured vessel producing an aneurysm. Blood will spill out of the vessel and pour into the brain tissue. You could have a metabolic source of disease. So, for example, you can have blood toxins from renal failure, or you could have diabetic ketoacidosis, or you can have an absence of vitamin B12, which is required for myelin formation. So if you lose myelin, you will lose communication between neurons, which will transform into loss of sensation or movement. You could also have sources that are electrical in nature, for example, epilepsy. And there also is one called paraneoplastic. There are not many examples of paraneoplastic. The example I give here is one that is the most common and serves to explain this type of disease. So, for example, you could have disease in the nervous system that is not primary. It's not the primary cause, it's a secondary cause. So it's secondary to a neoplasm or cancer that's outside the nervous system. An example would be a breast tumor. So a person has a breast tumor, and the normal reaction of the immune system when it sees abnormal cells is that it tries to kill them. So it develops antibodies for the abnormal breast tumor and attacks the breast tumor. Now, in some cases, the antibodies, which are designed to match the outside of the breast tumor, well, actually, it also fits well on other tissues, for example, the cerebellum. So the antibodies will bind to the cerebellum, and the immune system will start destroying the cerebellum. And so that will lead to loss of motor control because the cerebellum is responsible for motor control. You could have multifactorial sources of disease, and that simply reflects the fact that there are several factors that cause the disease, and an example would be migraines. Many things cause migraines. Sometimes we say the disease is idiopathic because we don't know the cause, and autism would be an example of a problem of neural function for which we do not know the source. So those are all the sources of disease. Now, the most common ones that would be seen by a family doctor would be migraine, stroke, Alzheimer's disease, traumatic brain injury, epilepsy, autism, Parkinson's disease, spinal cord injury, Tourette syndrome, multiple sclerosis, cerebral palsy, amyotrophic lateral sclerosis. So these are the most common. Most common seen by a family physician. Now, the family physician would refer to a neurologist for taking care of the patient, but the patient would first most likely see a family physician. Now, we will cover most of these diseases in greater detail through this part of the course. The treatments are very difficult for the nervous system because neurons do not repair themselves, and oftentimes disease kills neurons. So oftentimes the treatment for disease to the nervous system is to try to limit the effects or remove the source of the disease and try to promote normal function of the nervous system as much as possible. Occupational therapy is often used to help the person learn how to do things differently so that they can regain their normal daily routines. So we will now start looking at some of these in greater detail. The first that we'll look at is the vascular system. It's important to note that the brain is highly active, and neurons will die after 5 minutes without blood supply. And that's because the brain never stops. Actually, if you block blood supply to brain tissue, the brain would actually increase its activity because it's going to try to figure out a way to solve the problem. So that will actually make things worse. This is in comparison to the heart. The heart will only get damaged after 20 minutes without blood supply. And that's because the heart will slow down if it loses blood supply. Skeletal muscle can be fine for 6 hours without blood supply, because if there's no blood supply to the muscle, you won't be contracting that muscle. That muscle will not be working. And so it doesn't need much blood supply. So a main problem with the nervous system is that it never stops. It's highly active. And within 5 minutes, neurons will die. Oftentimes, when the activity of the brain is going to increase damage, clinicians will put the person in an induced coma to reduce brain activity so that they can allow mechanisms to heal the brain and so that the increased activity doesn't occur and damage the brain more. So if there's a problem with circulation of blood, we talk about stroke, and that will produce a loss of function. Now, there's 2 types of strokes. You can have a hemorrhagic stroke, or you can have an ischemic stroke. So hemorrhagic stroke would be a vessel that ruptures and the blood pours out of the blood vessel into the brain tissue, while an ischemic stroke would be blockage. And so the blood flow cannot continue. Now, I'm not going to describe to you all of the blood vessels. I'm just going to describe the circle of Willis. So these are the arteries that provide blood supply to the brain. And there's a very nice feature produced by the circle of Willis, which is in the center. And I'll make a blow-up of that area. So the circle of Willis is right here. So if you follow the cursor, you will see that I'm following the path of the circle of Willis, which forms a complete circle. So if you were small enough to walk inside the blood vessel here, you'd be able to turn or to walk all the way around. And that has quite a bit of an advantage for protecting the nervous system. So let's say, for example, that a person has complete blockage of the left internal carotid. So this here is the left internal carotid. And if we block this blood vessel feeds 2/3 of the brain. So you would expect there to be a very large damage. Normally, this blood vessel feeds through the middle cerebral artery and also the anterior cerebral arteries. So it's going to feed 2/3 of the brain tissue. But actually, nothing happens. What will happen if there is total blockage at this level here of the internal um, uh, um, carotid artery? What will happen is that the blood on the other side, which is the right internal carotid, it feeds through here. And normally it doesn't cross because there's pressure coming from the other side. But now there's no pressure because the blood vessel was blocked. So the blood will just come to the other side and start feeding the anterior cerebral artery, and it will come here and feed the middle cerebral artery and return flow to the brain. So there won't be any damage. That's the protection provided by the circle of Willis. In rare cases, you could actually have damage to both internal cerebral carotid arteries. And so you might expect that 2/3 of each half of the brain would be damaged, but actually nothing happens. And that's because the basilar artery here, normally it only feeds the posterior third of the brain. Normally it can't come up here because of the pressure provided by the internal cerebral internal carotid arteries. But in this case, since they're blocked, the blood will come up here and feed the middle cerebral artery and the anterior cerebral artery on both sides. So it will return blood flow to the brain. Now, the blood flow, it can't maintain as high a blood flow, but it can maintain sufficient blood flow for function. So there are many very old people who might have complete blockage of the internal carotid arteries, and they function normally, except that they get tired fast. So if you showed them your wedding photos, they might get tired after looking at 2 or 3 photos, because the processing of information to understand what's on the photos would exhaust them. They don't have the blood flow required to sustain an analysis of those photos. So that's the advantage provided by the circle of Willis. If the blood flow is blocked beyond the circle of Willis, so for example, if the blood flow was blocked here, that's the right middle cerebral artery, then the circle of Willis can't do anything. The blockage would prevent all of this area here to lose blood flow, and the tissue would die. So now let's look at more details of the types of strokes. So this is a picture that shows an aneurysm. So this is through an angiogram, a cerebral angiogram. And what happens here is that an artery, they put an opaque dye inside an artery. And so when they do x-rays, they can see the dye inside the arteries. So here there's bulging because the wall of the artery has thinned out. So normally there are several layers of muscle to withstand the high pressures inside an artery. So the pressure can go up to 180 or 200 inside an artery. So normally the arterial wall is muscular and can withstand that. But if there's damage to the muscle layers, then the wall will bulge out. And this can actually burst and bleed into the brain tissue, which we say parenchyma. And that would be a hemorrhagic stroke. So an example of a hemorrhagic stroke is shown here. This is actually a patient who died after taking cocaine. So most likely the patient had a weak blood vessel, and after taking cocaine, which is a very strong stimulant, if you take cocaine, your blood pressure would rise very high. And so the high pressures most likely burst the aneurysm, and the blood went into the brain tissue or the parenchyma. This is an example of an ischemic stroke. And in this case, it's a blockage of the blood vessel. So you could see here a thrombus, that is a blood clot that has formed somewhere. A blood clot forms inside an artery and then flows and gets caught when the arteries get smaller. So here the blood clot got stuck. And so downstream, all of the tissues would lose oxygen. Now, we talk about specific areas. We talk about the area close to the blood vessel, which is the infarction zone. So you can see this light colored area close to the occluded vessel as being an infarcted zone. This tissue will die. Now, on the either side of this infarcted tissue is what we call the penumbra. And so, for example, in this area here, this is part of the penumbra. And this area is between life and death. So it's losing oxygen because it normally receives oxygen and nutrients from that occluded vessel. But it also normally receives some from this vessel, which is not occluded. So this area here is between life and death. And this is the area that treatment is aiming to bring back to normal function. The infarcted area, after 5 minutes, the neurons will be dead and there's no recuperating them. So typically what the treatment is to give thrombolytic agents. Now, the thrombolytic agents are only given if it's within 4 to 5 hours of the occlusion, so of the stroke. That's why when doctors hear that a patient has had a stroke, they want to know when it occurred. Because if it's beyond 4 to 5 hours, the thrombolytic agents may have no effect or they might make things worse. So they will not give it. But if it's less than 4 to 5 hours, then they give thrombolytic agents, which will circulate in the bloodstream. They will digest the thrombus, and so then the blood flow will return, and the penumbra will heal, and the patient will regain function. So oftentimes, with the treatment, we see regain of function within 24 hours. If it's regain of complete function, then we talk about a transient ischemic attack. Otherwise, there might be some long-lasting effects, some effects that take a longer time to recuperate. It might take months, and in some cases, some loss of function will be permanent. The next figure shows an example. So you see here an example of an ischemic stroke. The person had an occlusion of the middle cerebral artery. We can tell because damage to the middle cerebral artery will lead to infarction of this area here. That's shown in the next figure. So the corresponding diagram is here. What was lost in the previous figure is this area here, and this area is supplied by the middle cerebral artery. Damage to the posterior cerebral artery will lead to damage here, and damage to the anterior cerebral artery or occlusion will lead to damage in this area here. So we can tell by the area of damage or the loss of function. In this case, it will be the face and arm, whereas when it's the anterior cerebral artery, it's mostly the leg. So through the loss of function, we can tell which artery was damaged. This figure is to describe the effects of calcium overload. The figure seems extremely complicated, but I'll go through it and you'll see that it's not too complicated. Essentially, what happens is that with ischemia, whether it's a hemorrhagic stroke or ischemic stroke, whether it's due to an aneurysm or a blockage, it doesn't matter. What happens is that neurons are lacking oxygen and nutrients, so you get ischemia. So the ischemia will lead to loss of oxygen or hypoxia in the cell. So the mitochondria, which use oxygen, will fail. And the mitochondria have several functions. One function is to produce ATP, energy for the cell. So the cell is going to run out of energy. And this energy is very important for removing calcium from within the cell. Calcium is a second messenger which triggers a lot of chemical reactions. So it's important to remove the calcium. But in this case, it won't be able to because it lacks the ATP to remove it. So calcium will accumulate and produce an overload. The mitochondria also has the function of sequestering calcium, that is to remove calcium. So if it can't do that, that also leads to calcium overload. Now, the ischemia leads to loss of function, and the brain is going to try to figure out a way to solve this. And to try to solve this, it needs to increase neuron activity, which means it's going to communicate. And the main neurotransmitter used for communication is glutamate. So there's going to be a lot of glutamate released. But glutamate causes opening of NMDA channels that allow calcium to enter the cell. So that's going to make the calcium overload even worse. So you've got this huge calcium overload, which is going to trigger a lot of chemical reactions. You're going to have a lot of chemical reactions, a lot of chemical products that are ready to be reacting with other products. And those are called free radical production. And this will eventually lead to cell death. Now, let's say that it's beyond 4 to 5 hours and the circulation is returned. So you reperfuse the tissue. Well, you're going to be providing oxygen. And the oxygen that was missing for these free radicals to really work properly is now available, and so that will actually kill the cells even quicker. The reperfusion will also bring the immune system cells to the tissue. They're going to recognize that cells are not normal. They're going to attack them and finish them off. And so that will lead more quickly to cell death. So this is the problem that occurs with calcium overload. This is a good spot to end this first lecture. So I'll stop at this moment here. Thank you very much, and I'll see you next time.